Dear friends and students, very good morning. In this video lecture, I will be explaining about the neurotransmitters and their pathways. First of all, I would like to give the definition for neurotransmitters. So, in neuro junction or in the neuroeffector junctions, some chemicals are liberated. So, these chemicals are called neurotransmitters. The neurotransmitters released either in the neurojunction or in the neuroeffector junctions. A number of neurotransmitters which is present in our body. Some of the neurotransmitters are excitatory in nature, some of them were inhibitory in nature. Some neurotransmitters have both excitatory as well as the inhibitory activity. So now the neurotransmitters, I said, either excitatory or inhibitory. So each neurons generally synthesize and releases a single type of neurotransmitters. For example, dopaminergic neurons releases only dopamine. Adrenergic neurons releases only noradrenaline. Cholinergic neurons releases only the acetylcholine. So they are all very specific in natures. So there are 7 important processes involved in the actions of neurotransmitters. First of all, the pre-requirement for a neurotransmitter is there should be some synthesizing enzymes for synthesis of neurotransmitters. Neurotransmitters has to be released only when the nerve is stimulated or not. There should be some agonist and antagonist available. There should be some enzymes for its rapid removal, and the neurotransmitters should act on the receptors. So these are all pre-requirements of a neurotransmitters. The 7 processes involved in the neurotransmission and its subsequent actions are: the first one is neurotransmitter molecules are synthesized from precursors under the influence of enzymes. So there should be some precursor. For example, for acetylcholine, choline plus acetyl-CoA. For noradrenaline, it is amino acid phenylalanine, etc. The neurotransmitters molecules are stored in the vesicles. Here in this figure, we can say these are all the precursor and these are all the enzymes responsible for its synthesis. So neurotransmitter which is formed are stored in the vesicles. If you see the electron microscopic structure of neuron, there will be a varicosity. The varicosity contains number of vesicles. In these vesicles, the neurotransmitters are stored. So the neurotransmitter molecules that leak from their vesicles are destroyed by the enzymes. There are some degrading enzymes which is present in the nerve. So after depolarization and generation of the action potential, the vesicles comes in contact with the varicosity. So these are all the vesicles that comes in contact with the varicosity. This is for the action potential and there will be release of neurotransmitter. This process is called the exocytosis. The released neurotransmitter molecules bind with autoreceptors. So that inhibits subsequent release of the neurotransmitters. If a particular amount of neurotransmitters is accumulated in the biophase, the further release is inhibited. The released neurotransmitter molecules can able to combine with the postsynaptic receptor. So these are all called the postsynaptic receptor, which is there in the effector organ. The released neurotransmitter molecules are deactivated either by reuptake mechanism, this is reuptake mechanism, or by the enzymatic degradation. So these are all 7 important processes in the action of neurotransmitters. So some of the neurotransmitters which is present in the body is acetylcholine, Then this neurotransmitter is used by spinal cord neurons to control muscles and by many neurons in the brain to regulate memory. The deficiency of acetylcholine leads to amnesia. Increased concentration of acetylcholine leads to Parkinsonism. So in most of the instances, acetylcholine is excitatory in nature. Because it produces contraction, it increases all the secretions of our body. The next neurotransmitter is dopamine. It is the feeling of pleasure. Dopamine has multiple functions depending on where in the brain it acts. The dopamine is usually inhibiting neurotransmitter, so deficiency of dopamine leads to Parkinsonism. The most important inhibiting neurotransmitters of brain is GABA, gamma amino butyric acid. Most of the CNS depressant like phenobarbital, barbituric acid, diazepam, alcohol, neurosteroids, they act on the GABA receptors. Of course, picrotoxin is also acting on GABA receptor. The next neurotransmitter is glutamate. It is excitatory in nature. The glycine is a neurotransmitter used mainly by neurons in the spinal cord. There are some specialized cells in the spinal cord called Rancho cells. The Rancho cell secretes glycine as the neurotransmitters. Another important neurotransmitter is norepinephrine, or is called noradrenaline. It is both neurotransmitter as well as a hormone. And this is responsible for the fight and flight response. It regulates normal brain processes. It's also involved in the depression and also antidepressant. The next neurotransmitter is 5-hydroxytryptamine. This is responsible for mood, appetite, and sensory perception. But at the level of spinal cord, serotonin is inhibiting pain pathways. The properties of neurotransmitters already I have discussed. These are all synthesized in the presynaptic neurons, localized in the vesicles in the presynaptic neurons, released from the neuron under the physiological condition called depolarization and action potential, and they are all rapidly removed from the synaptic cleft either by uptake mechanism or by degradation with the help of enzymes, and presence of receptors on the postsynaptic neurons. The binding of this neurotransmitter, the corresponding receptors elicit a biological response. For example, for acetylcholine it is muscarinic and nicotinic. For adrenaline, noradrenaline it is alpha and beta. For dopamine it is D1, D2, D3, 5-hydroxytryptamine 1 to 7. GABA, GABA A receptor, GABA B receptors. For NMDA, NMDA receptors. So the neurotransmitters are combined with its specific receptor and elicit the biological response. The neurotransmitters are acting on two kinds of receptors. One is inotropic receptors, where the involvement of the sodium ions, calcium ions, potassium ions, and chloride ions. The second receptor is a metabotropic receptors, where the activation of the G protein and involvement of the second messengers. Let us discuss how acetylcholine is synthesized and degraded in our body. So I said there should be some precursor for synthesis of neurotransmitters. In case of acetylcholine, it is acetyl-CoA and choline. Acetyl-CoA is formed from the metabolism of carbohydrate to pyruvate to acetyl-CoA. So the acetyl-CoA enter into the neurons with the help of choline transporters by facilitated diffusion. Then acetyl-CoA plus choline, with the help of cholinestyl transferase, there is formation of the acetylcholine. That acetylcholine is stored in the vesicles with the help of ACH transporter. So when this neuron undergoes depolarization and action potential, the vesicles comes in contact with the varicosity and the release of neurotransmitters. The neurotransmitters can able to combine with the corresponding receptors. When it undergoes metabolism, it is converted into choline plus acetic acid. This choline is again utilized for the synthesis of acetylcholine. The next neurotransmitter is dopamine, which is involved in the Parkinsonism. The deficiency of dopamine leads to Parkinsonism. The precursor of dopamine is tyrosine. Tyrosine is nothing but hydroxyphenylalanine. Phenylalanine is an essential amino acid which is converted into hydroxyphenylalanine. That is converted into dopa with the help of tyrosine hydroxylase. That dopa is converted into dopamine by removal of COO group. The reaction is called decarboxylation. We see some of the dopaminergic agonist and antagonist. This is the dopaminergic neuron where synthesis of tyrosine converted into dopa, dopa converted into dopamine. That dopamine is stored in the vesicles. Then after action potential, the release of dopamine. Now, the dopa synthesis can be increased by L-dopa, levodopa, which is used in the treatment of Parkinsonism. There are some drugs called reserpine and tetrabenazine, which is said to be catecholamine depleters, which interfere with the vesicular storage of dopamine. The stimulation of release of dopamine in the nerve terminal is promoted by the amphetamine, which is a CNS stimulant. Blocking of dopaminergic receptor and autoreceptor is with the help of antipsychotic phenothiazines and butorphanol derivatives like haloperidol. By blocking the dopaminergic receptor, they produces major tranquilizing action. The inhibition of the reuptake mechanism is by means of amphetamine and cocaine. So the dopamine concentration in the biophase is increased by amphetamine and cocaine. The dopamine is metabolized by an enzyme called monoamine oxidase. That enzyme is inhibited by the pargylene. So these are all some of the dopaminergic agonist and antagonist. Next, we will discuss about the neurotransmitters or hormone, which is responsible for the fight and flight. Is a sympathetic neurotransmitter. Is nothing but norepinephrine or noradrenaline. If your drug have or neurotransmitter have the catechol nucleus, that is dihydroxybenzene nucleus, that is called catecholamine. Under this catecholamine, we get the dopamine, norepinephrine, epinephrine, isoprenaline, etc. So the synthesis of noradrenaline is from the phenylalanine, then hydroxyphenylalanine with the help of tyrosine hydroxylase. So this tyrosine is converted into dopa. Dopa is converted into dopamine with the help of the pyridoxal phosphate as the coenzyme. The third step is again hydroxylation reaction where dopamine beta hydroxylase can hydroxylate dopamine and convert it into norepinephrine. The norepinephrine, when undergoes methylation, it is converted into adrenaline. So the basic difference between adrenaline and noradrenaline is presence or absence of methyl group. Noradrenaline do not have the methyl group, whereas adrenaline have the methyl group. So this adrenaline or noradrenaline is acting on the adrenergic receptor, that is alpha and beta. The next neurotransmitter is the 5-hydroxytryptamine, which is involved in the obesity, depression, migraine, and also the addiction. The precursor for 5-hydroxytryptamine is again the amino acid called tryptophan. When tryptophan undergoes hydroxylation reaction, it is converted into 5-hydroxytryptophan. 5-hydroxytryptophan on decarboxylation converted into 5-hydroxytryptamine. These 5-hydroxytryptamine agonists and antagonists are used for the treatment of various disorders. For example, it may be an antiemetic, it may be used for the treatment of migraine. The selective serotonin reuptake inhibitors are used for the treatment of depression. So that we will discuss in the separate lecture. The next major amino acid neurotransmitters are glutamate, which is excitatory in nature. GABA and glycine is inhibitory amino acid neurotransmitters. Now the glutamate is synthesized from the glutamine and which is packaged into vesicles glutamate. As I said earlier, depolarization and action potential leads to fusion of vesicles with varicosity, and there will be release of the glutamate, that glutamate combined with particular receptors. The next is GABA, gamma aminobutyric acid, which is the major inhibitory neurotransmitters of our body. It is found in every region of the brain virtually. Glycine is the main inhibitory neurotransmitter at the level of spinal cord. This is synthesized from glutamate with the help of glutamic acid decarboxylase and vitamin B6. The glutamate is synthesized. That glutamate is packaged into the vesicles, and this is removed from the synapse by the specific transporters in the glia and neurons, and this is mainly metabolized by GABA transaminase. The next neurotransmitter is glycine, which is major inhibitory neurotransmitter at the level of brainstem and spinal cord, which is synthesized from serine, taken up from the synapses or high affinity transporters. The glycine receptors. The drugs which act on the glycine receptor is the strychnine, and there are some selective bioactive peptides which can also be said to be neurotransmitters. For example, hypothalamic releasing factors, pituitary hormones, gastrointestinal and brain peptides, circulating peptides like angiotensin, bradykinin, opioid peptide like endorphin, enkephalin, dynorphin, neurohypophyseal peptides like oxytocin and vasopressin, neuronal and endocrine peptides like atrial natriuretic peptide, calcitonin gene-related peptide, and vasoactive intestinal peptides. With respect to opioids, the endorphin, enkephalin, dynorphin are the neurotransmitters which is involved in the pain response. They inhibit the flow of pain informations to the brain. These are all endogenous opioids. Morphine mimic these endogenous opioids. They are acting on the mu, kappa, delta, sigma, and epsilon receptors. The next neurotransmitter is substance P, which belongs to a family of proteins called the tachykinins. The substance P is nothing but polypeptide, which is made up of 11 amino acids. These are all acting on the receptors called neurokinin 1, which is found in primary afferent nerve endings, substantia gelatinosa and substantia nigra. And substance P is involved in the transmission of the pain, inflammation, nausea, mood, depression, as well as the migraine. The next neurotransmitter is nitric oxide, which is synthesized by nitric oxide synthase in neurons as well as in the blood vessels. The help of a endothelium-derived relaxing factor, they can dilate the smooth muscles in blood vessels. This is a mechanism of action of organic nitrites and nitrates. They can combine with the superoxide anions to form peroxynitrite, an oxidizing free radical that can cause DNA fragmentation and lipid oxidations. So with this, we have completed the major neurotransmitters of our body. In next lecture, we will discuss some other chapters. Thank you.